Assalamualaikum everyone. Welcome to another discussion on our series on clinical laboratory instrumentation. What we are interested in today are cell counters. We're going to structure our talk today with some motivation behind why we're interested in these cell counters. Then we're going to look at one specific type of cell counters called hemocytometers. We'll understand what hemocytometers are, when did they come into being, how did they function, especially the process of counting these cells using hemocytometers. We're going to look at some examples regarding that as well. And then towards the end, we're going to try and draw a comparison with the automated cell counters and see what's the benefit of using these automated cell counters as compared to the traditional hemocytometers. So, moving on towards the motivation behind this. So the motivation is essentially derived from a great many applications that these hemocytometers are used in. So you could, for instance, be interested in monitoring the health of the cells. You might be interested in seeing how the cells behave over time. Their transformations could be important for a particular study. Sometimes you're growing a cell culture. Um, or maybe there's a subculturing within those cells and you're recording cell growth over time, or the way cells behave in certain um, circumstances. So there are different applications regarding investigations of how cells behave, and you might be interested in different analysis. So for that reason, it's important to understand the morphological nature of these cells. Often we are only interested in cell count, and sometimes we're interested in how the cells look like. So we will see what hemocytometers give us in terms of information, but these are certain important motivations behind study of hemocytometers. Um, Also, experimental reproducibility, the fourth point, is also very important in terms of choosing the cell concentration in such a way that you want your experiments to be reproducible. And hemocytometers allow us to ensure that the cell concentration that we take of a sample is measurable. And when it's measurable, we're able to reproduce those experiments or standardize them for certain applications. So in terms of experimental reproducibility, hemocytometers play a very key role to standardize or ensure that similar concentrations or same concentrations are kept for same kind of experiments. Right, so what we are going to do now is look into some of the history before we further develop our understanding of these. These have been credited to Sergei Alfaro way back in 1884 with the initial designs or initial implementations of the modern day hemocytometer. And then towards the late 19th century, Louis Charles Molasses has been credited with bringing up significant improvements. What you're seeing now is a 1923 patent filed by Eugene Bruker, and again, Eugene Bruker is also credited with a specific design and making significant, important improvements in what the original designs of hemocytometers were. So enough about history. Let's understand what these basically are. So what is it that we're looking at? This rectangular sort of a rectangular dish or a rectangular slab with further rectangles in them. What are we looking at? We're basically looking at a, if I have to formally define it, it's a counting chamber device. It's used for counting blood cells. Hemo is blood in Greek, so if I have to break hemocytometers, literally hemo is a Greek word meaning blood, and cyto is for cells, and meter is to something that's for use for measurement. So it's blood counting measurement device. or blood counting device. You're measuring blood cells, you're literally counting them. So it largely consists of a glass microscope slide, and there's a rectangular indentation in the center, which creates a chamber and a space where blood could be pipetted in, or it could be deposited in, so that this slide could be seen under a microscope to literally count cells um, as you see them. So that's a very traditional way a hemocytometer works. You literally count the number of cells that you see on the slide. What this counting chamber does is it allows a uniform spread of the sample, so that the cells are they can be seen at ease under a microscope. So here's what they look like. I just want to show you a hemocytometer from a vendor. Important to note is the price tag that it comes up with. Obviously, there's a variation with different vendors coming up with different price tags, but this is what a typical hemocytometer looks like. Let's further look into the structure and build more on that. So you see a hemocytometer on the left hand side, and what you see on the right is a zoomed up view of this grid right here. So it's this grid that has been zoomed up on the right hand side, and whenever a sample is placed in here, you would be seeing the components of that sample, those blood cells, or those white blood cells, or the components. Actually, those particular cells would be visible for you to count. And then there's a criteria for counting. We'll go into that. But before that, what I want to focus especially is this V groove that you see on both sides. So what that does is it allows for you to place a pipette, the tip of the pipette right there, so that the sample could be loaded into these chambers. Typically about 15 microliters is used, but obviously it varies from different sizes of these hemocytometers, and from vendor to vendor it varies. So you would use a pipette to place the sample, load the sample in these chambers, and then you would allow it to settle down for about 2 to 3 minutes. You would allow this time so that the sample just settles down very nicely. You would prevent subtle or any sudden movements when controlling this hemocytometer dish. And if you leave it for too long, the sample might dry out. So you don't want to leave it out for too long. You don't want to do it too quickly. So about 2 to 3 minutes is recommended time to allow this sample to settle down. So and then there's a dish, a covering plastic dish that is slipped on top of this. It's not visible, but if you could make out these boundaries, perhaps there's a dish. It's probably visible around here. So that covering, the cover, it's called the cover slip, that is slid on top of this after the sample is loaded. Let's write it here. Cover slip. Yes. So once that is done, we have our sufficient time. We could proceed with the counting process. And the counting process comes with certain rules. And the rules for that are, we're looking at a grid, and we could consider perhaps these 9 large squares. we could further divide our grid into these 9 squares. So, and we could consider that as our reading. So once these 9 squares have been uh, designed, what we are then interested in is looking at the individual cells placed in them. The rules vary from certain labs to labs, and the standards also vary, but let me define certain uh, parameters for our case. Um, One of the rules could simply be that a cell, let me draw the cells first and then we will go over these rules. So I'm going to draw some cells first in white. Now that you are familiar with these 9 grids, now you can visualize them. We're going to go ahead and remove these. I'm going to go ahead and remove these 9 grids that I had marked. Yeah, so there we are. And we have our cells lined up here. What I'm also going to go ahead and do is put in some more cells in blue color, almost everywhere. So for one reading, what we are going to do is and this is what's typically done, is 5 squares are chosen for one chamber. So we could go ahead and pick these. These are standardized most of the time. So we will only be reading or counting cells in these 5 chambers, the 4 corner chambers and the 1 central chamber. So we would be counting cells in these 5 chambers, and the total number of cells that we're going to get at the end of the count will be our cell count. Before that, you notice there are 2 types of cells in this particular specimen. There are blue cells and then there are white cells. One of the reasons for that is we typically use a trypan dye. A trypan dye penetrates through the membranes of the dead cells and it colors them blue. So it's a blue dye, it colors them blue when you visualize it. The dead cells are going to come up as blue. So that allows us to differentiate between the living cells, which we call viable, and the non-living cells, which we refer as so the living cells are referred as viable and the non-living cells are referred as non-viable, and they appear as grayish and blue. I've drawn here as white, but they typically would appear as gray and blue, grayish, and the non-viable would appear as blue because of the trypan dye. The second rule, so when counting the number of cells, we would count all the cells, but when we have to differentiate between them, we would refer to them as viable cells and non-viable cells. The second important rule that we will take into consideration is that what happens when a cell is touching a particular boundary of a grid. So for example, if this cell right here is touching a boundary of this grid, and it's also touching boundary of the grid at the right beneath it. So should you count it or should you not count it? Because remember, we were interested in these 5 squares. So since we were interested in these 5 squares, what do you do with the ones that touch the boundaries, right? This is another example. So part of it is in the grid that we are interested in counting, and part of it is outside of that grid where we're not supposed to count. So what do you do? Should you count or should you not count? That is the question. So for that reason, we go ahead and we define certain rules, and I'm going to lay out those rules now. The rules could be as simple as this, that for cells that touch the boundaries in top and left side of the chamber should be counted in, whereas if they're touching boundaries on the bottom and right of this chamber of the squares, they should not be counted. So if I were to draw it, this is how it would look. So cells that are touching these red boundaries, these pink boundaries, they would be counted in for that particular square. But if they're not touching these boundaries, if they're touching any other boundary, they would not be counted in. So that's one of the rules that would allow us to successfully accurately count cells. So we're going to go ahead with some of the calculations now. Let's actually go ahead and count the white and the viable cells and the blue non-viable cells, and then let's build on our calculations from there on. So I'm going to go ahead and write down the actual number of cells for each of these blocks. So for this particular chamber, let's just write viable and non-viable. The viable cells that I see are in white, which are 1, 2, 3, 4, 5, 6, and the non-viable is just 1. Remember, I'm only counting this, not this, because it's on the bottom boundary, and we only count cells in top and left boundaries. So this has not been counted, which is why the non-viable cells equal 1 in this grid. Similarly, I'll go ahead and write for this 3 viables, 1 non-viable. For the central, 4 viable, 2 non-viable. For the one at the bottom right, we have 5 viable and 2 non-viable. For the bottom left, we have 1, 2, 3, 4 viable and 1 non-viable. Interestingly, if you note, this has been counted in because it's hitting the top boundary of the grid that in question, the grid we are counting in. So that is why it has been counted. So we have a total count right here. What we're going to do next is we are going to go ahead and count and sum all of them up together. Let me just sum up the viable cells. I'm going to call it as V total and NV total. Let's just do a total of these. So let's count the viable ones first. 6 plus 3, 9, plus 4, 13, plus 4, 17, plus 5, 22 viable cells. In the non-viable cells, there are 2 plus 1, 3, plus 2, 5, plus 1, 6, plus 1, 7. So I have my count of viable and non-viable cells. What I may be interested in next is perhaps percentage of let me write it down here. What I may be interested in next could be percentage of viable cells, and I know I have 22 cells that are viable divided by the total of 29. times 100. So this would give me a percentage of viable cells. Right, so these are certain parameters that we are usually interested in. We may also be interested in counting the average number of cells. We may be interested in average number of cells, and the average number of cells could be defined as viable cells per number of squares, which is 5 in our case, times 100. So if it's not a percentage, you would probably not want to time it with 100, but remember, this is average number of cells that you're interested in, so don't confuse it with the total number of cells, 29. When we're talking about average number of cells, we only consider viable cells. So that is 22. That should equal 22 over 5 as the average number of cells that we're interested in per square. So it should be per square. Average number of cells per square. So that is another parameter that we are typically interested in. The third parameter that we usually care about is the dilution factor, which equals basically the total volume of what has been pipetted into these chambers. It's a ratio with the cell volume, the volume of the cells that was pipetted in. So why is there a difference between total volume and cell volume? Cell volume is the volume of the specimen of the cells that you pipetted in. So that could be perhaps 100 microliter. And then remember, you added a dye, a trypan dye, to colorize your non-viable cells. So for example, that was also 100 microliter. So your total volume turned out to be 200 microliter, and the volume that corresponded actually to your cells was 100 microliters. So your cell factor turns out to be 2. A dilution factor turns out to be 2. So that is another parameter that we are also interested in, so, and dilution factor, right? So and finally, the last and perhaps the most important parameter that we're interested in is concentration. What is the concentration of the cells in our study? So that is one key parameter that we're interested in, and it's a combination of a few things. Let me write it down. I'll write it down here. So concentration is basically a product of average number of cells per square, which we computed, multiplied by your dilution factor, multiplied by your volume in each square. So we are aware of what the average number of cells per square is. We already calculated that, 22/5 right here. We know what the dilution factor is. We have computed that as well. And now we're interested in volume. Volume is the volume of sample that is represented by one square. For instance, what is the volume represented by this particular square on? We have 9 big squares that we initially talked about. So what's the volume represented by one square? Why I'm interested in one square? We are interested in one square because, see, we are using average number of cells per square. So this calculation for concentration is for each square. So it's representative of each square. So since we're using average number of cells per square, we are going to use volume represented by one square. And say the volume for each square was 10 to the power minus 4 microliters. Right, so if we go ahead and compute that, because this is just an example, I'm going to go out and let you know that this volume is 10 to the minus 4. It could be different depending on different standards. However, concentration is calculated in, because dilution factor would not have any units. If you look at its formula, the microliter gets cancelled with microliter. Average number of cells per square would have units of cells, right? Because we're counting cells. And the volume is essentially microliters, but microliters is in denominators because it's the volume for one square. So the units for concentration would turn out to be cells per, it's typically written in milliliters, but you may write it in microliters for simplicity. Whatever the volume is given to you, try to use the same units when you're calculating concentration. Right, so that is what that is pretty much it as far as cell counting on a hemocytometer slide or a chamber is concerned. You get to see exactly how many cells are there, and you get to count them, and then there are certain parameters that you have to evaluate. So how many parameters did we look at? I think we looked into average number of cells per square. That's one. Percentage of viable cells, that's another. We looked into dilution factor, and then we looked into concentration as well. So we looked into all 4 of these parameters, and then of course we could count the number of viable and non-viable cells as well. So that being said, there's one additional note in here, and that is sometimes cells tend to clump together, and then it becomes subjective to the viewer whether or not you would count them as one cell or 2 cells. So that's the discretion of the viewer. And sometimes the viewer might want to count them as one single cell, or they find that to be as 2 cells clumped together, and they seem as 2 cells, so they would count them as 2 cells. So that is very subjective, and it's dependent on the user. The other important thing is sometimes the cells are deformed, and when they're deformed, their shape is more or less slightly disturbed, and it's a bit arbitrary, a bit deformed. So you really find it hard to classify them as a viable cell. So you might want to classify them as a deformed cell, and keep a separate count of them rather than counting them as viable cells. So if they don't have a regular shape as the other viable cells, you might want to call them as a deformed cell and keep a separate count of them. Alright, let's now move on. Alright, so you're now encouraged to go ahead and take this as an example question to figure out those 4 parameters that were earlier calculated. So try and solve for these. Remember these the basic rules that were set up, that you're only going to count those 5 squares that we counted as and defined them in our rules, and we were interested in the cells that we are going to count those cells which are going to touch the boundaries on top and left for each of those squares. And also you would notice that in this particular example you have 2 kinds of cells, the circular ones that are circle and then they become elliptic and then ovalish, and then there are triangular ones as well. So this has been made simpler for you. Instead of color schemes, I've used 2 different shapes. So the triangle ones are non-viable cells, the circular and the elliptical or the oval ones are all viable cells. So go ahead and compute those 4 parameters that we earlier used in the example. Use the same volume used in the example and give it a try. And then we're going to discuss this at the end. Right. So now moving on to the next part, which is the automated counters. Automated counters are different kinds of counters. They come in 3 fundamental implementations. We're going to briefly look at that. The first ones are the image-based counters. So these image-based counters, they utilize a bright or fluorescent, they use a bright field. They use they basically employ certain dyes to ensure that microscopes are able to look at them. So they would be seen basically under fluorescent microscopes, and they're coupled with some digital cameras, which could be CCD or CMOS sensors. And the idea is to take images of those slides that you see under the microscope. And then those images could be fed to a image analysis software, and that software would then be counting those cells for us. So instead of a human counting those cells individually with a counter, trying to make those judgments, you would employ a software algorithm that would probably be making contours on each of these cells. And since they are dyed, they fluoresce, you would be able to probably use some vision-based algorithms utilizing their color information as well to separate the viable from the non-viable cells. So you'd be using a software. So these are essentially image-based cell counters. The next kind of counters are electrical impedance-based counters. So these are also called the Coulter counters, derived from the person who invented these. So the Coulter counters, they are particle counters. They're based on electrical impedance. Basically, as the sample, the liquid, the specimen, which contains the cells, is drawn through a small opening, as it moves through a small aperture, it's going to cause some change in the electrical impedance from that aperture as it's going to move. And we are constantly looking at measuring that impedance through that aperture. And as those cells are going to move through the aperture, there's going to be a fluctuation, a change in impedance. And that change in impedance is going to be proportional to the size of the particle. And using that information, these kind of counters, they could provide the number of and even size of the cells as they move through the aperture within the sample. But there's a drawback with this kind of implementation, is that they're counting the number of cells that pass through based on the impedance, but they are unable to distinguish the viable cells from the non-viable cells, as both would have certain impedance. So this kind of implementation would not be able to tell the difference between the viable and the non-viable cells. The third kind of implementation is the flow cytometry implementation of these automated cell counters. So you have a single cell stream passing through a laser beam, and the absorbance from that laser beam is measured. The scattered light is also measured at different angles, and we use that information to determine the cell's granularity, its diameter, and its morphological nature, to be so to speak. So you're able to tell, and this reminds me, it's pretty much similar to what we studied in scanning electron microscopes, where we had a beam, an electron beam hitting at the sample and then being recorded at different sensors to understand what the physical nature of the specimen was. So a similar phenomena here, where you're understanding what's the cell's granularity is, what is the morphological setup of the cell, its size, diameter, and that information is encoded in how the scattered light is recorded after going through that through those cells. So that's pretty much it from the automated cell counters. Here's one from a vendor. I would also direct you to go ahead and look at the user manual for this, or at least the brochure for this particular automated cell counter. It will be available in the description of the YouTube link. Go through that and answer a very basic question: that what kind of automated cell counter is this? How is this automated cell counter? What is the working principle of this automated cell counter? What is the implementation? We have just gone over very briefly in the previous slide, so try to figure out what this is doing. Also, look at the price tag for these automated cell counters and compare that with the hemocytometer that we had, which came in fairly cheap as compared to this price tag. So that is it for this discussion today. We're now going to follow up with our question and answer session, and we're going to go over the two problems that we have put up in this module, one of which was the cell counting and the other was trying to find what the implementation for this automated cell counter was. Thank you and good luck.